Hi everyone, and thank you Stefano and Isa for this opportunity to share some thoughts with this audience here at this important workshop. In the time that I have before you, I would like to talk about the threats to human health from waterborne diseases and examine very briefly how remote sensing can help in this context. I'm talking about water, and water is a giver of life. For life as we know on this planet, we need water for survival, for recreation, for culture, for food, for entertainment. But water is also like one of these Indian goddesses. It has many faces. When it turns on its destructive power, it can be very destructive indeed, and it can take away life where it once gave life. The problems that I'm going to talk about are huge. They are global in scope, but I cannot address it at that scale. So what I will try to do is to reduce it to a level where I can begin to understand what's going on, and hopefully I can also communicate some of that information to you. So for the rest of this talk, I'm going to be talking about this one lake, Vembanad Lake, in the south east coast of India. And these pictures are from a devastating furious floods that hit the lake and the surrounding areas in 2018. So the first requirement in the face of such devastation is to be able to map the flooded areas so that any follow-up action, remedial action can be targeted towards the areas that are worst affected. In this particular instance, Sentinel-1 product from ESA, provided through this agency IWMI, were used as a source of important information. This set of images are produced using microwave active SAR sensors. One of the limitations of such an approach is that these sensors do not provide information on a daily basis. So we also looked at complementing this information from visible spectral radiometry from Sentinel-2 data. And these are our results where we were able to identify water, no water, and flood waters. It's important in this kind of a study to emphasize the importance of local information. Sometimes what may appear to be floods may not be actual floods. They may be deliberate flooding of paddy fields for cultivation. So it's always important to build into these kind of remote sensing applications whatever local information we can provide to supplement the satellites. But these are examples of what remote sensing can do when there is, for example, floods. It could be drought. But outside of these extreme events, there is also an ever-present and invisible danger that comes from water. And I'm talking about vector-borne and waterborne diseases that require water for their survival at least on some phases of their life cycle, and which can then be the bearer of diseases. In the left side of the image, I have an image of a Vibrio cholerae bacterium responsible for cholera. Then there are the mosquito-borne diseases, and the mosquito life cycle requires a larval stage in water. If we take the particular example of Vembanad Lake, one of its problems is invasive species of macrophytes, floating vegetation, and water hyacinth, which is extremely beautiful. It is an invasive species in the lake. It creates stagnant pools of water and facilitates breeding of mosquitoes. So in a remote sensing perspective, there are now products referred to sometimes as floating algal index that you can use to map the extent of vegetation on the water. And if we can demonstrate that this kind of a product works for Vembanad Lake, then when we could explore its use for work related to malaria or dengue fever or any of these vector-borne diseases. But even more clearly, with some new satellite images, such as the one I show here, where the ground resolution is 3 meters, we can actually practically see the algal vegetation. So we can begin to look at the distribution of the algal floating vegetation and see how they are affected, for example, by the circulation and the man-made controls on the circulation. In the middle of the image is a dam that was built a few decades ago to help farmers, but it has also changed the circulation patterns completely. And you can see that the north and southern parts of the lake are quite different from the point of view of water quality. As part of an ongoing project called Revival, along with our colleagues from India, we have been carrying out extensive field sampling in Vembanad Lake, where we have looked at the concentrations of pathogenic Vibrio bacteria in the water and its their association with phytoplankton, with zooplankton, with the benthos, and with water itself. Of all the cases that we have looked at, the strongest associations were found between the pathogenic bacteria and phytoplankton concentration. So in this particular example, we have a couple of diagrams showing the environmental Vibrio cholerae presence or absence, and this is expressed as a probability, as a function of chlorophyll concentration. What's so striking in this picture, and it was unexpected, is that the brackish water in the north of the lake, brackish water, that's correct, in the north of the lake, and fresh water to the south of the lake, the relationships are quite different between phytoplankton and the cholera bacteria. We suspect that this difference has got something to do with the changes in the community structure of phytoplankton, and we are looking at this more closely. So if we have such relationships between phytoplankton concentration, or more precisely chlorophyll concentration contained in phytoplankton, then we can use satellite images of chlorophyll concentration to map the environmental risks from cholera, as shown in this example here. So that's one avenue that we are looking at. But remote sensing applied in this context has no predictive power. We get an image today. In the best example, we can produce a risk map for today. But it's also important to look at methods by which we can look forward in time, see if we can predict risks. And for this, we are employing epidemiological models of the type shown here, where we look at the infection in humans as a function of sanitation conditions, the aquatic environmental Vibrio conditions, the growth decay conditions that might determine how the environmental bacterial population might change with time, and how people are using drinking water, cooling, for water for drinking, cooking, bathing water. Key question is, are the people using lake water for these applications? And are they boiling the water before they use it, and so on. So this somewhat complex picture can be reduced to a series of model equations where we look at the susceptible population S, infected population I, and the recovered population R around the lake. And in the particular example that we have, or we are in the process of developing, and we have a student working on it. Her name is Libby Gould. The vulnerability to the disease is modeled as a function of phytoplankton and its relationship to the environmental concentration of Vibrio bacteria, the pathogenic bacteria. And when such a model is then combined with a Monte Carlo model, a Monte Carlo Markov chain model to fit the parameters, this is the type of relationship that we obtain between observed cases of cholera-like illness in the area around Bembenaad Lake and the model itself. Now, what goes into the model is lagged chlorophyll concentration with over 100 days lead time. That means that is the lead time that we have in the prediction as well. But let me emphasize that these are preliminary results right now. So what have we learned? Clearly, remote sensing offers several ways in which we can explore the dynamics of pathogenic disease-bearing organisms in water. But the problem is complex, and it is beyond any single group to be able to address the entire problem in all its complexity. So we need an integrated approach, and we need contributions and expertise from a variety of specializations, from scientists, from health workers, social workers, and so on, to work together to reduce the risk from waterborne diseases. We have learned from very intensive and extensive field sampling over more than 1 year in the Bembenaad Lake that we are always short of good data. So we need more data, we need good data, both clinical and environmental. Community engagement is key, and citizen science is an important avenue to the solution. For lack of time, I have not dwelt too much on that aspect of our work. The problem is global, I've repeated that, and it's not limited to the lake that I've been discussing. So we are in the process of building a global network of interested scientists to learn from each other and to work together to find common solutions and to find solutions that work in multiple locations and for different conditions. And what next? We are right now discussing another project on waterborne infectious diseases and global Earth observations. with ESA, and it will have, as envisaged now, 3 main work packages. One dealing with coastal areas and climate in relation to sanitation and environmental conditions. There is another work package that will deal with building data sets of all the relevant information, environmental and clinical, and then combining with modern statistical and other tools. I have listed here epidemiological models, artificial intelligence, and big data to understand what the data are telling us. Even though these modern methods such as artificial intelligence are all the rage, we feel that the epidemiological models that look at the functional relationships between these different boxes and pools is important to improve our understanding. The third and final strand that we have to pursue to make further progress in this field is capacity building and public engagement, perhaps through citizen science. And we have been talking with some of the participants at this workshop, notably from Médecins Sans Frontières and the World Health Organization. And at this point, I would like to stop and say thank you very much, and I wish this meeting all success.